Welcome to the Age Management Medicine Conference. We are here in Tucson, Arizona in 2017, and it's quite exciting because the field of precision medicine and genomics has arrived. Some folks like to think of it as personalized medicine. I'm delighted you're here. I know how excited you are about genomics and longitudinal analysis and looking at N of 1 data. So I have a few questions for you, and I'd first like to introduce Dr. Hans Röst, and you are assistant professor currently at the Terrence Donnelly Center for Cellular and Biomolecular Research in the Department of Molecular Genetics at the University of Toronto. Mm-hmm. I understand that's a new position because you've come from Dr. Mike Snyder's laboratory at the University of Stanford, where he's director of personalized and genomic medicine, correct?
Yes.
And a lot of your research emanates from there.
Yeah.
So we had some interesting questions for you, and the topic is going to focus on the notion of personalized omics data beyond the genome as informative for precision medicine and health, and how we keep our health instead of declining into disease.
Yeah, so I think one of the important things to understand with genomic data is that it's just one part of the picture, right? And your genomic data is going to be the same no matter what your age. So if you derive a risk profile from genomic data and you get information about what you're at risk at, you get the same answer if you're a baby or if you're an 80-year-old person. But of course, the kind of actions you want to take will differ very much on your environment and on your age. So we need to have additional data to actually understand what your current risks are for specific diseases, right? And we can only do that if we get additional data on top of the genomics data, and this is really where I'm interested in to gather this additional data. And a lot of the data we are looking at is the microbiome, it's transcriptomics data, it's proteomics data from the blood, and metabolomics data from the blood. And what we are hoping is that bringing these two types of data together, the genomics with the dynamic data we can acquire throughout the lifetime of a person, that we can get a much better insight into the actual current disease risk, and that we can also observe how a person transitions either towards disease or actually gets better and stays healthy over their lifetime.
Wow, that's beautifully said. I love it. And I think that points to the fact that genes are in fact mostly static. They're not dynamic. And what you're really speaking to is genes precipitate change in the body. DNA goes to RNA, goes to protein and molecular changes. And the fact is we can work on those changes as we live our lives, whether it's changing the way we eat, the way we work out or we don't work out, the kind of supplements or medications. And perhaps by detecting and predicting that at the basic level, even in utero or certainly beyond, we can change the trajectory of how we experience life. Maybe stay vital till the very last moment, maybe live till 102 if we can prevent the onset of disease or delay it.
Yeah, definitely to understand how far disease has currently progressed and to what degree we can halt the progression of disease or even reverse it, we need to look at this dynamic data, because that information will not be present in the genome. And this is especially true for metabolic diseases. So the only exception is cancer, where you can have a different genome in the cancer genome itself. But for most other diseases, the genome is static, right? And we need to look at this dynamic kind of data on top of the genome.
Absolutely. I think we started with, even within cancer now, precision medicine, we've come to understand how specific each cancer might be to the human being hosting it. And even though a cancer might be labeled lung or prostate or breast or brain, it might behave completely differently in different individuals, and targeted therapies have now come to be understood as the way to go. Somebody might need chemo, other people might need radiation. Certain drugs might be good for one person but not good for another person. Yeah. And the same is true of other interventions that would be important to learn. So tell me, in doing this, why is it interesting to us here? And I know we're both very interested in N of 1 data and using a unique individual's information, starting with a baseline and perhaps collecting it every decade of their life, beginning even as a young person in their teens or 20s for sure, when we're physiologically at our peak, and then perhaps being able to follow that person, using that person as their own force, as their own control.
Yeah, this is definitely something we have seen in our own data, that we just observe a lot of interpersonal variation between people. So there is a lot of genetic and environmental effects that at the moment we do not fully understand. So we do not fully understand why some people have, relatively speaking, high or low levels of certain analytes in the blood. And if we had full information about, would understand everything about their environment and their genome, we might be able to understand that. But at the moment, we are not at this level yet. So what we can do is we can measure their analytes in the blood and then compare them relatively over time, and then understand, well, how do they change relative to how they were when they were 20 or 30 years old. And this is really what we have seen also in our research, that you get much more powerful statements and also much more statistical power if you compare people to themselves as their own reference.
Exactly. That's fascinating, because another point you make is that it allows you to cut down on the number of subjects while you collect that. So by looking at a person as their own control, instead of perhaps having 1,000 people in a study, in order to get valid information, you may have 20, and you're going to extrapolate that data to show change over time because you know all the variables around that smaller group.
Yeah, and it's important to work with the right statistical tools, but it's inherent that if you have fewer confounding factors, which you do have in a longitudinal setup, that you don't need as many people as in a cross-sectional setup. And this is really what we have found also for larger biomolecular studies.
Fabulous. Well, I can't tell you how excited I am about that, since I started on that path 20 years ago, possibly before you were born. I'm not sure. Maybe not quite. But anyway, that's fabulous, and I look forward to all that data coming forth. What can we learn about longitudinal data? What have you learned from the work you've done over the last few years that you're hoping to continue in Toronto?
Well, I think we are still very much at the beginning of understanding how this longitudinal data can help us understand disease and disease risk. So I think what we are now understanding is just about how important and powerful this kind of data can be, and how it can help us to understand what's going on during the lifetime of a person or during a specific perturbation or intervention. And this is definitely what we will be looking at going forward in our studies. Absolutely.
Well, I know that I was quite excited when I came across Mike's data himself. Professor Mike Snyder did his own genetic data, his own. He looked at RNA, he looked at the proteomics, he got the proteins, and he found himself becoming a diabetic when he caught the flu, not even recognizing that that was in his family, which indeed it was. And the same is true for what we collect. We recognize and predict and can detect disease, which as we monitor and intervene, we're actually able to reverse in the folks that we take care of. And so it's quite exciting time in medicine, and I'm hoping to see that brought to fruition in my lifetime yet, where we're actually taking care of people and treating and preventing disease before they actually get sick. Makes sense?
Yeah, I think that will be the optimal.
That sounds like a real healthcare system as opposed to a sick care system.
Exactly.
Good.
Yeah.
Well, it's exciting. Good luck in your next stage of your career and on becoming a father.
Yeah, thank you very much.